Now it's time to discuss about a process of spermatogenesis. To know about a process of spermatogenesis, in general, let us see what is gametogenesis. Gametogenesis. Gametogenesis is a process where haploid gametes are formed from diploid gamete mother cell. Diploid gamete mother cell in gonads. That is gametogenesis. A process of formation of haploid gametes from diploid gamete mother cell in gonads. Gonads means reproductive organs. It is injury. It is of two types. First one, spermatogenesis. We have to define in the same way. Spermatogenesis is a type of gametogenesis where haploid sperms are formed from sperm mother cells. Sperm mother cell, diploid sperm mother cell in testes. Testes are male gonads. Spermatogenesis is a type of gametogenesis where haploid sperms are produced from diploid sperm mother cell in testes. And sperm mother cells are spermatogonian cells. Spermatogonian cells. In a similar way, second type, oogenesis. Oogenesis. Oogenesis is a type of gametogenesis where haploid egg or ovum is formed from egg mother cell, a diploid egg mother cell in ovary. Ovary is a female gonad, is a type of gametogenesis where haploid egg or ovum is formed from diploid egg mother cell or ova mother cell in ovary. This is oogenesis. And egg mother cells are oogonial cells. Oogonial cells. Gametogenesis is of two types, spermatogenesis and oogenesis. And spermatogenesis is with respect to male reproductive system. That is a process of formation of haploid sperms from diploid sperm mother cell, that is spermatogonial cell in testes. Today our work is to study about a process of spermatogenesis. A process of spermatogenesis. A process of spermatogenesis. First we have to define it. It's a type of gametogenesis where haploid sperms are formed from diploid sperm mother cell, that is spermatogonial cell, in seminiferous tubules of testes. Seminiferous tubules of testes. And now this process takes place in 4 steps. 1. Period of multiplication. Second is period of growth. Third is period of maturation. And the 4th step is Spermiogenesis. Spermiogenesis. These are the 4 steps. First one, period of multiplication. Second is period of growth. Third is period of maturation. And finally, spermiogenesis. This process takes place in seminiferous tubules of testes. Seminiferous tubules of testes. In the lining of seminiferous tubules, already we know that 2 types of cells are there. Most of them are spermatogonian cells. Along with them, a nourishing serotoli cells. And now let us see those spermatogonial cells. Let me draw one spermatogonial cell diploid in nature. Spermatogonial cell. That is there as a lining of seminiferous tubule. In first step, a period of multiplication, these spermatogonial cells undergo mitosis. So that their number is increased. Their number is increased. As mitosis takes place, they are diploid only. In the first step, period of multiplication, by mitosis, spermatogonial cells get increased. Why the number of spermatogonial cells should be increased? We already know that sperm production takes place in terms of millions. In terms of millions. That's why we need more number of spermatogonial cells. So that in the period of multiplication, spermatogonial cell undergo mitosis, their number is increased. And these are also spermatogonial cells only. In second step, period of growth, these spermatogonial cells, their cellular and nuclear content are doubled, increased. That is the period of growth. The cellular content as well as nuclear content are doubled, increased in these spermatogonian cells. Let me go with a single spermatogonian cell. This first step is a period of multiplication, and second step is period of growth. Both cellular as nuclear and nuclear content are increased. Now, such spermatogonial cells are named as primary spermatocytes. They are named as primary spermatocytes. Now they are ready to undergo maturation. That is third step. And all the spermatogonial cells, their cellular and nuclear contents are increased. They become primary spermatocytes, but they are diploid. And now, in the third step, that is period of maturation, these primary spermatocytes undergo meiosis. But we know that meiosis takes place in two steps. Meiosis first and meiosis second. Meiosis first is reductional division. There is a reduction in chromosomal number, so that in the first step of period of maturation, meiosis first takes place. As meiosis first takes place, from this primary spermatocytes, two cells are formed. As meiosis first is reductional division, these are haploid in nature. These are none other than secondary spermatocytes. Secondary spermatocytes. By meiosis first, a single primary spermatocytes can give two equal sized cells. Those are named as secondary spermatocytes. As meiosis first is taking place, as it is reductional division, these secondary spermatocytes are haploid in nature. And now these two undergo meiosis second. Meiosis second is just like mitosis, so that from each secondary spermatocyte, two equal sized cells are formed. From one secondary spermatocyte, two cells, and another secondary spermatocyte, two cells, and all of them are haploid only, because meiosis second is just like mitosis. These are also haploid, means from them the cells formed are also haploid. And now these are named as spermatids. Spermatids. And these two steps is named as period of maturation. Period of maturation is taking place like this. And the final step, spermiogenesis. These haploid spermatids, they are oval in shape. In the final step, that is spermiogenesis, these spermatids are metamorphosed into haploid sperms. From this haploid sperm, from this haploid sperm, yes, sperms are produced. And this step is named as Spermiogenesis. Hence the formation of haploid sperms from diploid spermatogonial cell is taking place in 4 steps. First one is period of multiplication. During that step, spermatogonial cells number gets increased. Why the spermatogonial cell number should be increased? As we know, sperm production takes place in terms of millions, so that we do have more number of sperm mother cells. That takes place in period of multiplication. And the second step, all these spermatogonial cells undergo a period of growth, during which their cellular content as well as nuclear content are doubled or increased. And then those spermatogonial cells are named as primary spermatocytes. Now these primary spermatocytes undergo period of maturation that takes place in 2 steps. Those 2 steps are meiosis first and meiosis second. When primary spermatocytes undergo meiosis first, 2 equal sized cells are formed, but they are haploid because meiosis first is a reductional division. And now these haploid cells are named as secondary spermatocytes, and both the secondary spermatocytes undergo meiosis second. From each secondary spermatocytes, 2 equal sized cells are formed. As meiosis second is just like mitosis, there is no reduction in chromosomal number. Once again they are haploid as well, and they are all named as spermatids. These 2 steps, including meiosis first and meiosis second, is a period of maturation, and the final step is a spermiogenesis. Spermiogenesis is a final step where spermatids are metamorphosed into haploid sperms. Haploid spermatids are metamorphosed into haploid sperms. That process is said to be spermiogenesis. Remember, a main process is spermatogenesis. The definition for spermatogenesis is a formation of haploid sperms from diploid spermatogonial cell in seminiferous tubules of testes. And what is spermiogenesis? Is a final step in spermatogenesis, where haploid sperms are formed from haploid spermatids. This is the way they can ask, even distinguish between spermatogenesis and spermiogenesis. That spermatogenesis is a type of gametogenesis. Spermiogenesis is a final step in spermatogenesis, and in spermatogenesis, haploid sperms are formed from diploid spermatogonial cell. In spermiogenesis, haploid sperms are formed from haploid spermatids. And once in NEET, the question was asked, which of the following is odd one out? Which of the following is odd one out? Where they had given 1 primary spermatocytes, 2 secondary spermatocytes, 3 spermatids and spermatogonian cells, primary spermatocytes, secondary spermatocytes, spermatids and sperms. These four answers were there. This one, second, third and a fourth. They were given. The question is, which of the following is odd one out? Primary spermatocytes, secondary spermatocytes, spermatids and sperms. Logically, we may think in such a way, these are all immature sperms and these are completely formed. So that we may name this is absolutely wrong. If you just see their nature, primary spermatocyte is diploid and these three are haploid. Then which among them is odd one out? Primary spermatocyte is odd one out as it is diploid, but these three are haploid. And now while dealing with that male reproductive system, I have explained a process of spermatogenesis takes place when the males reach puberty or adult age. Their adult age starts when they become 14 years old. During that, hypothalamus produces gonadotropin releasing hormone. This is produced from hypothalamus. Gonadotropin releasing hormone produced by hypothalamus. Once this gonadotropin releasing hormone is produced, this stimulates anterior part of pituitary. This hormone stimulates anterior part of pituitary so that it produces two hormones. One is LH, another one is FSH. LH is luteinizing hormone and FSH is follicle stimulating hormone. LH luteinizing hormone, FSH follicle stimulating hormone. This LH stimulates Leydig cells. They are there in interstitial space. Once LH stimulates Leydig cells produce androgens named testosterone. This testosterone helps for spermatogenesis. This spermatogenesis process will be started by this testosterone. And this FSH stimulates Sertoli cells. FSH stimulates Sertoli cells which produce some factors, and these factors help for Spermiogenesis. And what is spermiogenesis? This FSH stimulates Sertoli cells. They produce some factors, and these factors help for spermiogenesis. Spermiogenesis in the sense, spermatids are metamorphosed into sperms. Final step. That final step is due to FSH, as it stimulates Sertoli cells to produce some factors, and that process is taking place. That's why you know that FSH is considered as gametokinetic factor. Gametokinetic factor. Which of the following hormone is said to be gametokinetic factor? That is none other than FSH. Not only in males, even in females. In females, egg or ovum is formed. That is due to FSH. In case of males, by this FSH, gametes are formed. Hence, the other name for FSH is gametokinetic factor. They can ask the question, which of the following hormone is said to be gametokinetic factor? That is follicle stimulating hormone. And once the sperm production starts in case of males from their puberty age, it goes up to their old age around 75 years old. There will be a sperm production continuously. And there will be a ejaculation at the time of coitus, sexual intercourse, that gives out 3 ml of semen. In this 3 ml of semen, 200 to 300 million sperms are there. In that 3 ml of semen, 200 to 300 million sperms are there. Among them, 60% of them gain normal size and shape. Among 203 to 300 million sperms, 60% of them will have normal size and shape, and 40% of them are having vigorous motility. They are moving very fast, 40% of them, because their movement is very much necessary. When the semen is ejaculated in the female genital system at the time of sexual intercourse, and the sperms have to pass through the uterus and they have to climb that oviduct and go to the upper part where they can fuse with the egg. That vigorous motility is also very necessary. Among the total sperms produced, 60% of them are having normal size and shape, and 40% of them are having vigorous motility. And I have told about that male sex hormone, one and the only male sex hormone, that is testosterone. Even it induces secondary sexual characters in case of males. Those secondary sexual characters in case of males are strengthful body, coarse voice, it means hardening of voice, and appearance of mustache and beards on the face. It's about a process of spermatogenesis. Now, let us discuss about the structure of human sperm. This is human sperm. Sperm is a haploid, matured and a very minute male gamete. And it looks like a tadpole larva of frog. It looks like tadpole larva of frog. And is a haploid, mature, very minute and motile, movable male gamete. And it looks like tadpole larva of frog. As far as its structure is concerned, its body is divided into head, neck, and this is middle part, and this one is tail. Sperm is distinguished, sperm body is distinguished into head, neck, middle part and a tail. Head is globose and is having a prominent nucleus, a large prominent nucleus with 23 chromosomes. It means one set of chromosomes. In that one set of chromosomes, there may be 22 autosome and X or 22 autosome plus Y. There are two types of sperms. Some sperms are having 22 autosome plus X chromosome, and other 50% of sperms are having 22 autosome plus Y chromosome. Due to this, human males are named as heterogametic. It means two different types of male gametes are produced. Human males are heterogametic, but human females are homogametic, where almost all the female gametes are having 22 autosome and X. There are only one kind of gametes, but here two types of gametes. And a nucleus with 23 chromosomes. In that 23 chromosomes, 22 autosome plus X or 22 autosome plus Y. Just above the nucleus, there is a C-shaped or cap-like structure. This is named as acrosome. Acrosome. And this acrosome is filled with hydrolytic enzymes. Hydrolytic enzymes. A hydrolytic enzyme present in acrosome is hyaluronidase. That enzyme is hyaluronidase, and that hydrolytic enzyme present in acrosome plays very important role at the time of fertilization with egg, because when head touches the outer membrane of egg, acrosome bursts and hyaluronidase enzymes spread over the outer membrane. They become softened so that sperm can penetrate into the egg through the membranes. For that, hyaluronidase plays very important role. That is there in acrosome. And with respect to acrosome, one neat question was asked. Acrosome present in sperm is formed from which of the following organelle? Acrosome is formed from which of the following organelle? Where they had given endoplasmic reticulum, ribosome, Golgi complex, lysosome like this. And acrosome of sperm is produced from Golgi bodies or Golgi complex. We should remember, acrosome is formed from Golgi complex. And now, total sperm is covered by plasma membrane because it's a cell. And in between head and middle part, there is a neck. And here we do see two centrioles. Two centrioles. One is proximal centriole. Another one is distal centriole. In the neck region, there are two centrioles. As it is an animal cell, two centrioles must be there. Those two centrioles together named as centrosome. Proximal centriole means it is present close to nucleus. Proximal. And a distal centriole means is away from the nucleus. From this distal centriole, there is a central axis which runs through the middle part, even in tail, and goes up to the end of tail. And this is named as axial filament. This axial filament is developed from distal centriole and passes through the middle part. It goes up to the end of tail. And middle part, it is totally packed with mitochondria. Mitochondria. They are the powerhouses of the cell. They are meant for releasing the energy. And this energy is used by the tail for its vibration. That helps the movement of the sperm in semen. These mitochondria, which are packed in that middle part, help for releasing the energy. They release the energy that is used by the tail. And as far as tail is concerned, it is distinguished into main piece and end piece. The tail is distinguished into main piece and end piece, and the main piece is having a protoplasmic sheath. Protoplasmic sheath. But the end piece doesn't have a protoplasmic sheath. And this is about the structure of human sperm. And the lifespan of human sperm is 24 to 48 hours. The lifespan of this human sperm is 24 to 48 hours. Within this, it has to fuse with the egg, hence the fertilization. It is about the structure of human sperm. While dealing with testes, we have mentioned there are 250 compartments in testes. Those are named as testicular lobule. In each testicular lobule, there are around 1 to 3 seminiferous tubules. Now it's time to study about a diagrammatic section or sectional view of seminiferous tubule. Here I have drawn one testicular lobule. This is one testicular lobule in which the seminiferous tubules are there. Totally, this is one testicular lobule. This is one testicular lobule. In each testicular lobule, there are around 1 to 3 seminiferous tubules. They are the structural and functional units of testes. These are seminiferous tubules. This is one seminiferous tubule. Even these are also seminiferous tubules. We know that there are two types of cells in seminiferous tubules. Those are spermatogonian cells. Spermatogonian cells, they are germ cells. They only undergo spermatogenesis and produce sperms. And another cell present in seminiferous tubule is Sertoli cells. Sertoli cells. They are present along with spermatogonial cells. And these Sertoli cells, they are meant for nourishment. They provide food to germ cells. And these spermatogonial cells, they are diploid in nature, and from these spermatogonial cells, by multiplication, by the process named mitosis, those spermatogonial cells get increased. That we have studied in the process of spermatogenesis. And those spermatogonial cells which undergo growth, where their cellular content as well as nuclear content are doubled, then they are primary spermatocytes. These are also diploid in nature. Primary spermatocytes, they are diploid in nature. They are produced from spermatogonial cells. And these primary spermatocytes, by meiosis first, they give rise to secondary spermatocytes. As the meiosis second is taking place, now reduction in their chromosomal number. From this, the next generation of cells in the seminiferous tubules are secondary spermatocytes. Secondary spermatocytes. And these secondary spermatocytes, which undergo meiosis second, leading to the formation of the marking. I have made a little mistake. Spermatogonial cells, they are in first row. Next to that is from them primary spermatocytes, and from primary spermatocytes there is a secondary spermatocytes, and when they undergo meiosis second, the next generation of cells is spermatics. These and secondary spermatocytes, they are haploid. Even spermatids are haploid. And these spermatids, by spermiogenesis, they are metamorphosed into haploid sperms. Haploid sperms. All these things we have studied in a process of spermatogenesis. And in the seminiferous tubules, we do see two main types of cells: maximum spermatogonial cells and serotoli cells. And those spermatogonial cells, they undergo the process spermatogenesis. Once they undergo spermatogenesis, from those spermatogonial cells, primary spermatocytes, secondary spermatocytes, and spermatids, and finally sperms are produced. Sperms are produced. Everywhere in all the seminiferous tubules, I have drawn those 5 series of cells started from spermatogonial cells to sperms, and here and there serotoli cells. And then in between seminiferous tubules there is a space. This space is named as interstitial space. This space is said to be interstitial space. In this interstitial space, we do see another type of cells. Those are Leydig cells, meant for the production of androgens, named testosterone. They are produced by Leydig cells, and as these Leydig cells are there in interstitial space, these are also named as interstitial cells. Leydig cells or interstitial cells, and these are meant for the production of testosterone. In addition to that, in interstitial space there are some immunologically competent cells are also present in this interstitial space. Immunologically competent cells are also present as well. This is a diagrammatic sectional view of seminiferous tubules. Seminiferous tubules. This is one totally testicular lobule, in which 1, 2, 3 seminiferous tubules. Within the seminiferous tubule, we do have 2 types of cells: serotoli cells and spermatogonial cells. Spermatogonial cells, during the process of spermatogenesis, giving a series of cells named primary spermatocytes, secondary spermatocytes, spermatids, and sperms. And this process is continuously taking place. That production is taking place continuously, right from the puberty age of males up to 75 years of old age. These sperms will be produced, and these Leydig cells or interstitial cells are there in interstitial space, and they are meant for the production of testosterone. But already we know that Leydig cells produce testosterone only when that LH, luteinizing hormone, stimulates Leydig cells. They produce testosterone. That's why you know that LH, luteinizing hormone in case of males is said to be interstitial cell stimulating hormone, that is ICSH. Other name of LH, that is luteinizing hormone, is interstitial cell stimulating hormone because LH in case of males stimulate that interstitial cells or Leydig cells to produce testosterone. It's about the diagrammatic sectional view of seminiferous tulips.